This is TWIS. This Week in Science, episode number 839, recorded on Wednesday, August 25th, 2021. Digging in the science soil. I hope we come up with some good science seeds. I'm Dr. Kiki, and today we will fill your head with isolation, food, and microbes. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. It's approved! The COVID vaccine gets government scientist seal of approval. And now hesitant Americans everywhere are finally rushing out to get vaccinated. Or so we hope. In reality, there are enclaves of Americans who have been left out of understanding science. They haven't been left out by science teachers or scientists or science news. They have been willfully kept ignorant of science by ideology. Ideology that does not like what science has to say. And so, unbiased rational thought is assaulted relentlessly, conditioning minds to see science as fraudulent, agenda-driven, and even nefarious. The desperate manner in which the unvaccinated now fill hospital beds to capacity is not their doing alone. It takes convincing. Misinformation, disinformation, all-out assaults on the sense of self-preservation. These aren't stupid people. They are people who have been lied to, repeatedly, continuously, generationally. They are victims of ideologies that lose power when exposed to reason, that lose credibility when science is actually understood, that punch holes in stained glass windows every time there's another episode of This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
Woo! We are all here. Good science to you, Justin and Blair, and everyone out there. Welcome to another episode of This Week in Science. We have a full show for you today. Not just vaccines for everyone who would like them, but science, much science. We have an interview this evening with Dr. Noah Fierer, and I'm going to continue to mispronounce his last name, and I'm going to apologize profusely throughout the show. But we will be speaking with him about soil microbes and their place in humanity, sustainability, and our world. Additionally, I have stories about the intersection of space and health. Brains, because brains are pretty awesome, and additionally, some social Tourette's. What do you have, Justin?
I've got American know-how, gardens of terror, cannibalistic papers, and prison profits following policy.
And alliteration. Blair, what is in the animal corner?
Alliteration, huh? I've got fat flies, babbling bats, and messed up monkeys.
Wow. Blair's alliteration corner.
It still has it's still the same acronym. It works. It totally works. Everyone, if you have not yet subscribed to This Week in Science, you can easily find us all places podcasts are found, as well as YouTube and Facebook. We are TWIS Science on Twitch and on Twitter and Instagram. Our website. Yes, you can find all this information at our website, TWIS, TWIS.org, twis.org. And now we dive in to the science for the week. Okay, let's start with space and health and oxygen. Oxygen is the key to the interaction between these two supposedly very disparate areas of science. This week, it was reported that thanks to the COVID pandemic, hospitals are using much more liquid oxygen than they usually do, and this is causing shortages of liquid oxygen that will affect the space industry.
Oh,
The rockets
wow.
that launch rely on liquid oxygen as a propellant, and so they have limited supplies of this propellant, and it will cause delays in many of the scheduled launches for SpaceX and probably ULA. ULA's CEO, Tony Bruno, he tweeted that they were having issues with their liquid nitrogen supplier, which is also important in the launching of rockets. But the liquid nitrogen supplier is busily using their liquid nitrogen to help make more liquid oxygen, because liquid nitrogen has a lower freezing point, boiling point than liquid oxygen. So as a result, they use liquid nitrogen to make more liquid oxygen. After they do fractionated distillation, basically they take air and then they boil it to steam, fractionate the different components of the air, because each of them have different boiling points. They each come out, and you have gaseous oxygen that can then be turned into liquid oxygen if you have liquid nitrogen. It's a very complex process. Not too complex, but it's a complex enough process.
That's fine.
Yeah, it's a complex enough process that this is
ruining space.
Yeah, it's a manufacturing bottleneck, and it could also affect public health, not just in our hospitals, but also with relation to water purification, because many water purification systems rely on liquid oxygen for their processes. This thing, COVID, it is the thing that brings together various areas in the sciences that you never expected would be connected.
Yeah, and not in a good way.
No, not in a good way either, for sure.
Oh boy.
Yeah. But anyway, that's more fun than talking about what's the new name of the vaccine that the FDA just approved? Comirnaty. They say it is a combination of the name COVID-19 vaccine, the term mRNA, and is meant to sound like community.
Yeah.
That is some good marketing speak if I have ever heard it. But congratulations to Pfizer for being approved by the FDA. And we hope that this new approval will lead many people to get vaccinated.
It's like nobody knows, or at least most people probably don't know the name of the pill that women take that prevents pregnancy from happening. It's just the pill. This is just the vaccine.
Well, you also don't know who made your flu shot either, right? You didn't say, which flu shot did you get? I don't know, and I don't care.
Yeah, but this is the one that's the vaccine. It's not this vaccine, that vaccine. It's the vaccine.
There are many the vaccines, though, and Moderna has now submitted all the paperwork necessary for their approval process with the FDA. So potentially there will be another ridiculous pharmaceutical name to remember in the future for deciding which vaccine for COVID-19 you would like to take. Justin, tell me a story.
Oh, okay. Oh, we did it! We finally did it!
What?
Congratulations, America. Most of you believe in evolution now.
When you say most, do you mean like 51%?
Hey, it's most. So it's actually, they're saying, according to this, that it's gotten significantly above the halfway mark. This is a study based on a series of national opinion surveys that were conducted over the last 35 years, 1985 to 2010, they say. There was a statistical dead heat between acceptance and rejection of evolution. This is according to lead researcher John D. Miller, Institute of Social Research, University of Michigan. But acceptance then surged, becoming the majority position in 2016. Examining the data over 35 years, study consistently identified aspects that led to this difference. Aspects of education, civic science learning, literacy, taking college courses in science, and having a college degree were the strongest factors leading to acceptance of evolution. And it says here that, according to Mark Ackerman, researcher at Michigan Engineering, almost twice as many Americans held a college degree in 2018 as they did in 1988. That's great news. It's hard to earn a college degree without acquiring at least a little respect for the success of science.
Yeah, many university programs have science for non-majors courses that do cover a lot of this information. I actually taught one of those biology for non-majors way back when I was in grad school. So the rounding of education for people who are attending public institutions can be, the importance of that cannot be understated for potentially reaching this point. But perhaps also the teaching of evolution in high schools has gotten better as well, in schools generally. Yeah. I don't know. It's interesting.
It's a wonder. It hasn't always, but it is. But here's the context that you also have to put this in. There was an international, a study, a survey of 34 developed nations back in 2005. That looked at the acceptance of evolution. Basically, it's one question. Human beings, as we know them today, developed from earlier species of animals. Do you agree or disagree? Kind of that simple. In 2005, the only country that scored lower than the United States out of 34 developed nations was Turkey. So even as I'm congratulating ourselves, yeah, it's still not, doesn't feel like winning. Now,
what is winning, right? What is your definition of success? We have reached one goalpost here. We've hit a marker. So now let's keep improving on this and iterating on what success means for the understanding and acceptance of the idea of evolution, the theory of evolution within the general population. Perhaps our goal now is not
to get lapped twice.
Yeah.
In the race.
Well, I think the other question that I have is if they actually do causally link college education and understanding science in this way, right? Then you really have to address the institutional issues with access to college. How much it costs, how different demographics have it easier to access that education than others. And I think that if that's really how we're educating people, is based on something that is not offered to everyone equally, that's a problem.
Yeah. Yeah, absolutely.
Yeah, so
great
we've
point.
We've reached 54%.
54%.
And there's some in the middle. It might be like, still don't know. I don't know. There might be some in the middle that just... current study consistently identified religious fundamentalism as the strongest factor leading to the rejection of evolution. Go figure. There's, if you can believe it, there is also a political divide.
I can believe that there would also be a bit of a political divide.
If you just look at
idealistic
liberal Democrats, 83%.
Interesting.
Believe in evolution. I know, like, who's the 17%, right? I guess like there's a group. Only 34. Well, there is. There's still people who maintain their religion who have find it coincides the rest of liberal democratic ideology, I suppose.
Absolutely.
There's also 34% of conservatives accept evolution, which is actually that's 8% improvement over the years. So
I'm curious what the age spread is.
It's not given here.
Yeah, but I think that would be interesting to see, because also is some of this just that there's different generations that have different percentages of the population now than did previously.
It could be, but it's also, if you look at that percentage, whatever it is more likely to have a college education now, which is also like the vastly, that probably runs with, right? Somewhere.
Yeah. In terms of demographics and how people are affected by things, Blair, you've got a study related to fruit flies.
Yes. Yes. I don't know. After the last 18 or infinity months, depending on how you want to calculate it, you'll feel a little sluggish. Maybe were you not sleeping well? Maybe were you eating like garbage? Maybe did you gain the quarantine 15? Does any of this apply to you? Well, it turns out some of this could apply to fruit flies. Scientists have found that lone fruit flies quarantined in test tubes sleep too little and eat too much after only about 1 week of social isolation. This is a new finding which aims to describe how chronic separation from groups leads to changes in gene expression, neural activity, and behavior in fruit flies. And so this provides one of the first robust animal models for studying the biological ramifications of loneliness. This is from the Laboratory of Genetics at Rockefeller University. Michael W. Young, who is a professor there, he found, quote, that loneliness has pathological consequences connected to changes in a small group of neurons, and we've begun to understand what those neurons are doing. So once again, the fruit fly, Drosophila, is a great test subject because they are social creatures like us. They feed in groups, they serenade one another, they have mating rituals, they tussle, and they, after all this, they sleep a lot. They sleep around 16 hours a day. They have a midday nap and then a full night's rest. Doesn't that sound nice? And so in this study, after 7 days housed together, these groups of varying sizes produced no anomalous behaviors. Even if you had two cut off from the crowd, they did fine. So if you're paired up in quarantine, if you want to draw those conclusions based on this fruit fly study, maybe you did better than someone who wasn't. But when a single fly was entirely isolated, the lonely fly ate more, slept less. And they found that this was a group of genes linked to starvation expressed differently in the brains of lonely flies. And this was a small group of brain cells known as P2 neurons. So they shut down the P2 neurons in some flies to see what would happen. It's kind of separate to see if this is what was happening in this kind of social experiment. The chronically isolated flies suppressed overeating and restored sleep if you shut down the P2 neurons. But if you boosted the P2 neurons, if they were also isolated, it caused them to eat and sleep as if they had been alone for a full week. So it was those neurons. They found the neurons responsible for this behavior. And they went into whole other things. They engineered theoretical flies to look at this further. But suffice it to say, there's a good kind of idea of why this happens, at least in fruit flies, could potentially lead to something that we see in ourselves. One of the possibilities for why this happens evolutionarily, which is always my question, right? Professor Young said that social isolation signals a degree of uncertainty about the future. So if you are suddenly alone and you are supposed to be in a group of whatever your species is, there's a really good chance you're about to get eaten. So you're supposed to get eaten, or you've been separated and you need to find your way to that group or a new group, or you don't have extra eyes to watch for predators. Or you don't have extra eyes to forage for food. And all that to means if you find food, you got to eat it, and you can't let yourself sleep too long. So
yeah, I was going to say how interesting that it's starvation neurons and, thinking about loneliness as social starvation. But if you take it to that evolutionary explanation, it's survival based. It's the uncertainty. It is the fact, okay, I don't have anyone. When am I going to get food next? When am I going to find another conspecific, another individual of my kind? I don't know. So I need to take care of myself. I need to be alert.
So you can actually be love starved.
Yes. Yeah. And, during the whole pandemic, there were a bunch of studies that said that good numbers of people were gaining weight and losing sleep throughout it. And they a lot of the times it was attributed to anxiety or stress or uncertainty.
But where did that come from?
Yes. So there was general stress and anxiety about the pandemic, but this shows that there might be a whole nother piece, which is an actual genetic source of some of this behavior related to isolation. So
our social nature hardwired into who we are potentially.
Yeah.
Fascinating.
Uh-huh.
And along the lines of food, not starvation, but actually maybe how to get yourself to eat a little bit better, some researchers have published in Nature Food their new evaluation of nutrition involving environmental aspects. So how much water does food use? What are the environmental impacts of the food that you're eating? So not just the nutritional health aspects, but the environmental health aspects as well. And they've created this new framework that they're calling the HENI, Health and Environmental Nutritional Index. And in this, they're able to classify different types of food as giving you more life hours and being beneficial to your life through nutrition and being good to the environment, or being detrimental and taking away from the environment. And in their assessment, the US diet that they looked at, 5,853 foods, they ranged from losing 74 minutes to gaining 80 minutes per serving of food. These analyses indicated that substituting, though, this is part of an interesting conversation about meat and red meat eating, where everybody says if you eat less meat, don't eat cows, don't eat the red meat. If you eat less red meat, that's the best thing you can do for the environment and your health. Well, they say it doesn't have to be an either or, you eat it or you don't, but they say substituting only 10% of daily caloric intake from beef and processed meats for fruits and nuts and vegetables and legumes and seafood could give you up to 48 minutes per person per day of healthy living, and a 33% reduction in your dietary carbon footprint. Just 10% less of those red meats a day if you were to substitute them.
This is never going to make it to food packaging because
it's too complicated.
No,
even
if you handed me a Twinkie and that Twinkie said like 1 hour of your life, I'd be like, I'm not eating this Twinkie. Are you kidding? That's an hour of my life.
Yeah, it's going to take away your life. And so they have a wonderful assessment of, these 5,000 some odd foods. But there are some foods that definitely reduce, give you many hours lost. And so foods like Frankfurter sandwiches, not so good. But on our food pyramid, these are probably already not or the food web, whatever we use nowadays, these are not recommended foods necessarily. But some of the best things you can eat, peanut butter and jam sandwiches. Who knew?
Really?
Yeah.
Oh,
if you put more of an emphasis on the peanuts.
Staple. Yeah. My childhood staple just added so much to my life. I had no idea.
Great. All those peanut butter and jelly sandwiches we all ate as kids, except for those of us who have peanut sensitivities, have added to our lives.
Or jelly aversion.
Yes. Yeah, you could just have a peanut butter sandwich. It would probably be great. This is This Week in Science. Thank you so much for being a part of this show. We hope you're enjoying it. And if you are, share it with a friend today. I would like to take this moment to introduce our guest for tonight. We are joined by Dr. Noah Fierer, and he is a professor in the Ecology and Evolutionary Biology Department and a fellow in the Cooperative Institute for Research in Environmental Sciences at the University of Colorado. He's also the director of the Center for Microbial Exploration. His lab explores the distribution and roles of microscopic organisms in diverse environments and the relevance of microbes to the health and function of ecosystems, plants, and animals. Dr. Fierer, welcome to the show.
Hello, thanks for having me on the show.
It's so great to have you here. Now, we've talked with many people about microbes through the years, but I think the direction that your research takes is very interesting. And specifically, one paper that I do want to touch on today gets at where there are no microbes, which is something that's very kind of this foreign idea. But first, I'd just love to know, how did you get into microbes? What started your journey and exploration?
Well, it's a good question because I don't think people, kids don't grow up and be like, I want to study microbes. Maybe now, actually. I mean, I think if we've learned anything from this pandemic, it's that microbes are pretty damn important, right? I love studying microbes, I will say. So my group looks at microbes in lots of environments, including soil and on plants, microbes in our homes, in the atmosphere. And I think for me, what's really fun about studying microbes is how little we know. It's not very hard to find really basic questions. Like, for example, if you take a tablespoon of soil from your garden, what microbes are in there and what are they doing? There's unlimited questions, even in that tablespoon of soil, that we don't have the answers to. So that's what makes it really fun, is it's not hard to find something that's potentially very important to study, but for which we don't have a good understanding.
Right. And what specifically, though, how did you get into looking at microbes in the soil versus, a lot of people now are interested in our interaction inside our bodies with microbes, the human microbe interaction. But why soils?
Believe it or not, soils are pretty fascinating and important, right? And often, of course, we take it for granted, right? And we're walking around on a lawn. We don't think about the soil underneath our feet. But they're important for, the health of ecosystems, for human health directly and indirectly, for growing crops that we eat, of course, for emissions of greenhouse gases. The list goes on and on. So, soils are really central to a lot of the ecosystems and inhabitants of those ecosystems. I started my graduate work. I got really interested in soils. And again, for the same reason for microbes, is that there's a lot of unanswered questions that basic things we don't understand about soils and who's living in those soils.
Yeah. So you mentioned greenhouse gas emissions, and the first thing that I think about there are the Siberian tundras or these other areas in the far north that are warming, and these icy frozen bits of soil that have been frozen for years and not active, suddenly they have microbe activity again. Like did we expect that to happen? Like this is this crazy effect of climate change.
Oh yeah. And it's, what's the extent of it? How much methane and how much CO2 is going to be released from these systems with warming, and where? It's not going to be evenly distributed. And what's the magnitude of it? Is it going to create a positive feedback loop, right? You get more CO2, more methane coming out of these systems, which increases the global warming potential even more, which then exacerbates temperature change. It's the stuff of nightmares, really.
These are not my nightmares. I try not to have these kinds of these kinds of dreams, but it is a reality. So you mentioned the differential between different locations. So Siberia, obviously, it was frozen. Did we expect there to be a lot of microbial activity when the tundras thawed? And did we expect there to be life in that frozen soil? Was there evidence prior that microbes could survive in cold, frozen conditions for a long period of time?
Oh, for sure. I mean, in the tundra system, to us, it seems like a really rough place to live, but to a microbe, it's great. Yes, it's cold, but typically a lot of these tundra soils, there's lots of organic matter, so lots of things for microbes to eat. So as soon as the temperatures are right, it may only be a few weeks a year when it's warm enough. It's like Thanksgiving dinner, right? They've got all this organic matter and they'll just chow down. And of course, one product, the microbes eating that organic matter in a tundra soil, for example, is CO2 and methane. These are the gases some of the gases that we're worried about.
So thinking about how the different microbes, what they eat, where they live, the kind of limits to what environmental conditions they can exist in. What have you started looking at, and where did you start asking questions about that?
Yeah, so my group has been looking at microbes in soils from all across the globe. We've worked in Peruvian Amazon, we've worked in deserts, we've even worked in Central Park, New York City, the exotic locale. And, not surprisingly, there's different microbes. So we're really interested in what sort of microbes are living in these soils. And then, of course, what are they doing? How are they affecting us directly or indirectly, and how they're affecting ecosystems? We've looked in lots of systems, and not surprisingly, microbes are everywhere, and they're highly diverse. Again, you can take tablespoon of soil and you've got thousands of different types of microorganisms in there, most of which there's no name for. We don't have a species name for them. We know they're there, but we don't know what they're doing, how they may be affecting plants or how they may be affecting us. We just know that they're there.
And we just want one named after the show. Somewhere.
We'll see what we can do.
But what you're bringing up is actually really critical, crucial information to have going forward, because as this global temperature is changing, as the areas where plants are growing now maybe not habitable anymore, we don't just get to pick up the plants and reseed them elsewhere. We actually have to bring the microbial community of the soil with them. Otherwise, they might not be able to sustain themselves.
Exactly. I mean, we know from a lot of research, for example, on the gut microbiome, that, we're not made to live in a sterile environment. We're colonized by all sorts of microbes, and those play a really important role in our health. And the same is true for plants, right? Plants rely on a lot of those microbes on their leaves and in the soil around their roots to survive, particularly under stressful conditions like drought or temperature, low temperatures, for example. There's increasing evidence that those microbes that are associated with plants are really important for the health of the plants.
How do you determine, since we don't know all the microbes that are in that tablespoon of soil in a various location, how do you actually go about determining who's there?
Great question. So oftentimes, not exclusively, but oftentimes we use DNA sequencing. And this is the same exact DNA sequencing that's used in, CSI shows, right? Or, for human genome analyses. The technology is actually the same. And what we're doing is we're just, extracting DNA from soil, we're sequencing that DNA, and we can say, oh, look, we found this type of microbe or this type of microbe. And most of them, as I said, they don't have a name. So I always find it funny when plant and animal biologists are like, we found a new species. They're so excited. I'm like, I'm excited when I find something that we do have a name for, right? Because most of them we don't have a name for.
Something we know, yes.
So when you're blasting this stuff out, though, I mean, is it just sort of falling into these big broad families, or is it stuff that's like, is there stuff that's sort of outside of that we really don't even have an origin story for?
Oh, usually we know, okay, it's related to these other things. So we can say, okay, it's in this phylum or in this class or in this order. And that's definitely true for bacteria. So we generally know, who it's related to, or we have an idea. I mean, there's always things where like, okay, here's an entire phylum. So this broad grouping of bacteria, and no member of this group has ever been described.
Wow.
So then, we have the DNA sequence for it, and sometimes we can even reconstruct the whole genome, but that's it. We can't grow it in the lab. We don't necessarily know exactly what it's doing. And then, of course, there's viruses in soil, and that's a whole nother ballgame because there's lots of viruses that live in soil. Most of them infect bacteria. And that's really terra incognita. There's just a lot of viruses that we know nothing about, and what are they doing in these environments?
They're the bacteriophages. They're the
Exactly.
viruses that are going after the microbes, right?
Exactly. Yeah.
Not just us.
Yeah.
Not
all microbes are bad, right?
Yes, not all of them are. Some of them, a lot of them, they're very interrelated, and they have an ecological function, and they're there. They have eked out a niche for themselves. So we've got these tablespoons of soil that you're going and picking up. You're doing DNA sequencing, sometimes being able to figure out species, sometimes not. You've got genes potentially that can give you functionality for what can you find out what these microbes are doing in the different locations and kind of figure out ecological profiles of that?
Exactly. Yeah, we can say, okay, well, it seems to have these genes, so let's make some guess about this is how it's, for lack of a better term, making a living in that soil environment. We also can say, okay, we only tend to find these in, tropical forest systems. We don't tend to see them in deserts, so they're probably not too tolerant to dry conditions, right? Stuff like that. We can generate some hypotheses, but to really study that, to really know, okay, this is what this microbe eats. This is what it does. This is how it survives in its environment. You often need to grow it in the lab, and that's challenging.
Yeah,
Most of the microbes in soil are very difficult to grow in the lab.
microbes in general have been difficult. There are several that love laboratory conditions, but the majority do not.
Exactly.
Yeah. So along the lines of going from to dry conditions, I'm going to take us now to Antarctica, which is not it's dry in a lot of places. It doesn't have a lot of
It's a dry
precipitation
cold.
necessarily. Yeah,
It's a
dry
desert.
cold.
There's less than 10 inches of rain a year. It's a desert.
Yes. And so your team went to Antarctica and took a bunch of samples from different places. What was the goal there, and then what did you find?
Great question. Yeah, so it was about 3 years ago. I had the opportunity to go with a team of other scientists to look at the soils of Antarctica. So we always think of Antarctica as being ice covered. And of course, a lot of Antarctica is ice covered, but not all of it is. So there's exposed areas, particularly in the mountain ranges, that have never been covered in ice for hundreds, if not thousands of years. And there's soil there. And that's what we were going after, is trying to look at what sort of biology is going on in these soils of Antarctica. And I will say it was an awesome adventure. It's my first time there, and it was incredible.
And what did you end up finding? I mean, I am jealous of the Antarctic adventure, but I'm sure it was more than frozen, very interesting.
Yeah, so we were working in a remote region of Antarctica in the Transantarctic Mountains. So Antarctica itself is remote, and we were even in a remote area in Antarctica, right? So in the middle of nowhere, camped on this glacier. We got to, fly around and collect samples from all these environments that had basically never been visited before. And again, these are soils, so these are exposed, they're not glaciated. They haven't been covered in ice in hundreds, if not thousands of years. And we collected about 250 samples. And then came back. We put them on the slow boat all the way back because they had to stay frozen for preservation. And they actually went back on an icebreaker all the way to California. And eventually they made it to Colorado in my lab. And I had a graduate student that looked at them. And I'm like, let's start seeing what sort of microbes are living in this unique environment. And I will say what he found was really surprising to me. And he found that
was.
I'm building up.
I can't tell you.
Building the suspense. Can you just read about it? It has an ending.
Not just kidding. What he found was that in about a quarter of the samples, there was no detectable DNA from microbes in there. Nothing. And again, we've gone all over the world. There's microbes everywhere, right? Especially in soil. Every soil we've looked at, there's plenty of DNA, plenty of microbes there. So of course, what did I say to him when he came back with those results? My student Nick. First thing I said is something went wrong.
Yeah,
check it again.
Try it again.
Exactly.
Nope.
And of course he did, because it was surprising to us. And there was plenty of soils, about 75% of the soils that we collected from Antarctica, where there was all sorts of there's unique microbes, and we found plenty of them in there. But there was that 25% where we couldn't detect any DNA. So he went back to the lab, probably begrudgingly and cursing me under his breath, and ran some additional analyses, and we coupled it with a wide range of other analyses, trying to grow them on Petri plates, trying to measure how much ATP. So microbes all produce ATP for energy. So measuring the ATP in these soils, and all the results were pretty consistent in that we couldn't detect any microbes in about 20 to 25% of samples from Antarctica. And again, these are old soils. They've been forming in place for long periods of time. They're not covered in ice. So this was quite surprising to us. And I do want to make it clear is we can't say they're sterile, because you can never prove a negative, right? There's always a chance there's some
you didn't pick something up. Yeah.
Exactly. Yeah, there could be some fungal spore that happened to drift in from New Zealand and land there, and we just didn't pick it. But we couldn't detect anything using techniques that were specifically developed for these very low biomass types of environments. So basically, we were bumping up up against the limits of life. Conditions that were so rough that even microbes couldn't survive there.
Wow.
That is amazing to me that we would have a place on our planet. When we talk all the time, in the news, it's always extremophile this, and the ability of microbes to survive in so many places. And We have evidence of microbes living on the exterior of the International Space Station, or at least surviving in a spore form. So how is it possible? What is the limit? Is it just temperature? Is it water? Is it pH? What environmental aspect could possibly limit the growth factors?
Well, what we think is happening is that those soils that we couldn't detect any microbes, so no bacteria, no fungi, no other microbes, it's sort of a unique combination. It's very cold, obviously, Antarctica. Don't need to tell any of your listeners that. Very dry. So some of these sites, based on looking at the geochemistry and the soils themselves, have probably never seen liquid water for hundreds of years. So snow may fall on there, but it's so dry, it just sublimates and never actually forms liquid water. And the soils are also very, very salty because they'll accumulate salts from the atmosphere, but because there's no liquid water, those salts don't go anywhere. They just stay there, and over extended periods of time, they accumulate. So what we think is happening is it's like a hat trick, right? You've got cold, salty, and really dry. And that combination just makes it really difficult for microbes to live there.
But it's probably the only, like, the best place, like we've been talking about Blair for a long time. That might also be the best place to find a life that didn't originate with the rest of life if there's open niches.
One thing that's super interesting is, now that we think we're seeing sort of these limits of life, then the next question, to me at least, is, okay, well, who's able to survive right up to those limits? What sort of unique ways of making a living, like how are these microbes able to survive like right up at the limit of these challenging conditions?
Yeah, and so I am seeing two really good questions in the chat room right now. So one is, so what's going to happen when the Antarctic heats up even just a little bit?
Oh, that's a great question, because one reason we wanted to study these systems is they are currently changing very rapidly. So, if we go back there 5-10 years from now, it's highly likely that these systems will be different. And to give one example, I said these systems are super dry. Like they haven't seen liquid water by our estimates in many decades. But you can imagine a little bit of warming on 1 or 2 days is just enough for a little bit of that snow to melt and form liquid water. And then all of a sudden you can remove some of those salts, conditions are a little bit better, and possibly some random spore that lands there might be able to grow a little bit. So you can imagine even a half a degree change in temperature could make this environment go from what we believe is currently uninhabited to still not a great place to live, but potentially inhabitable.
Slightly better.
If something can make their way into that niche and make it their own, they have no competition.
Yeah.
Right.
For now.
For now, right.
For now. That's how the game works. The other question in the chat room was about Mars. We talk about on the show all the time about how nothing can ever be truly clean when we send it into space, no matter how hard we try. But if we know the limits of where at least earthbound microbes can live, maybe that can help indicate if our microbes would be able to live on Mars, for example.
Exactly. Yeah. I mean, I don't know anything about Mars. Never been there. But one reason people do study soils in Antarctica is because it is believed to have some similarities to soils. There are soils on Mars, including the cold temperatures, the saltiness, and particularly some toxic salts and the dryness. So that's one reason people study Antarctica. I don't know what's on Mars, so I'm very hesitant to say, okay, well, based on this results, this is what we'll see. Right. But I think one thing it does show is if we're going to use methods to try to detect life in Mars, we should probably test them out in these environments first. Because Antarctica is not that accessible, but it's more accessible than Mars. And so, this is a nice system for really detecting or seeing if we can detect microbes or other life right at, sort of the limits of habitability.
And as we get to the limits of habitability, I'm wondering how we can also use what we're learning in this Antarctic situation to, I guess, create clean rooms for NASA. Can we take the conditions in Antarctica and say, okay, let's make it super cold. Let's make it dry. Let's suck all the humidity out of the air and see what happens. Can they kill everything on the surface of the next Mars rover?
Yeah, I think the scientists and engineers working on it would love those conditions. Right? I mean, I think one outcome is there's always additional methods we could use to really try to detect very small numbers of cells in an environment. And so we could take some of the methods we've used in Antarctica and we could apply them. In fact, they are being applied to clean room situations where how do you prove a negative? How do you prove that there's nothing there? And that's really hard. Because all of these methods have some limits of detection. So, and we're bumping up against that. So, yeah, I mean, we don't want to send microbe contaminated stuff to Mars. Like, that's not good. So really refining those methods. That bad?
Because, maybe, what if this
Justin is pro sending microbes to Mars. He has no issue with
Marsifest destiny. Yeah.
Well, he's pro bringing back the mammoths. He's pro filling all the niches with whatever you want.
No. The mammoth had a niche. It should be, but it's not going to be there long anyway. But no, what if this is our we're talking about as if there's going to be this future generation, which I still hope there is, that's going to terraform Mars and we'll have 2 planets. What if everything just shuts down? Like, say there's a global pandemic that uses all the oxygen. We can't launch rockets anymore. What if those microbes are the one thing that got to leave this planet and go to Mars? I don't know if I would limit too hard those cleaners.
Putting eggs in more baskets. I see what you're saying there. Yeah. In terms of the topsoil and how we're looking at these topsoil, so we're looking at, okay, here are the limits to microbial life potentially. Are you team bacteria, team fungus? If you were to expect a microbe of any kind to be the first that you would find after not having seen anything there, what do you think it would be?
I'll back up and say the term microbe is not a very useful scientific term, right? It just means things that are small, right? So, this includes viruses, it includes a protist like amoebae, it includes bacteria, it includes archaea, it includes fungi. And I guess I would say don't underestimate the fungi. Fungi are tough. In some of these soils where we don't see any bacteria, we do see some fungi there. So they're able to survive in this super cold, dry, salty environments. So my guess, if I had to make a guess, is the fungi are going to be the first ones there. Or very few select few bacteria.
Kiki, when you were talking about the some microbes are hard to grow in a lab, the one thing that's hard not to have grow at places you don't want it to is the fungi. The fungi will show up in other experiments where that thing didn't even want to grow. It just, you have to be super careful with how you handle those spores because they will proliferate like crazy.
Yeah, your home is filled with fungi. Filled.
So
when you're in your home breathing, you're just breathing in fungal spores all the time.
Woohoo! Yeah, I have heard fungal researchers who study infectious disease actually say that they're more worried about fungus in the future, fungal species, as it gets warmer and we see different diseases emerge from various places around the equator. They're more concerned about the fungal species than they are microbial.
No, there's a huge intersection that between understanding what's in the soil and how plants are going to interact. And there's a lot of effort in biotech right now to try to develop sort of cocktails of microbes that you can match with plants to improve the health of crops. Are there aspects of what you've been working on that kind of point to some of these things that look really promising?
Oh yeah, so it is a great question, right? So in one sense, we have these soils that have all this diversity, right? Like nature has given us this amazing diversity of bacteria and fungi and viruses. And then at the same time, we need to grow more plants on less land to feed people, right? So it's an obvious thing to be like, okay, we already know that many plants use fungi and they use bacteria to grow better. And nearly all plants that we know of, have these symbioses, these interactions with microbes. So then it's an obvious next step to say, okay, can we add particular microbes to soil or to seeds or onto leaves to make them resistant to disease, make the plants resistant to disease, to make them grow better under drought or more resistant or use less fertilizer, for example. And there's definitely some cases where that has been shown, where people have been able to do that, but it's a tough nut to crack for some of the same reasons I already mentioned, that there's so much that's unknown. And such a small subset can be grown in the lab. It's really hard to test how a bacteria, for example, might affect plant growth if you can't grow it and add it to plants.
Or you've discovered these 20 microbes will absolutely improve the health and resilience and the growth factors of this plant. And so we're going to put these 20 into the soil with how many other competitors
exactly.
and how many interactions? Oh.
Right, and just dumping it on there, exactly. It may not necessarily grow. It may not interact the way you want it to interact with the plant. I mean, it's the same exact thing in gut microbiome, right? The same exact thing happens. How do we introduce the bacteria we want into our gut without having negative effects? And how do we actually show which microbes are beneficial?
Yeah, according to all the advertisements, it's just eat yogurt. You'll be fine, right?
Yeah,
So
even
it's a
though
little
really
bit
you're
more
doing
complex.
what Justin talked about, you're just throwing like 5 microbes into a sea of a community. They're like, who are you? Get out of here.
Yeah, there are some very interesting studies at play there. But I wanted to talk very quickly before we finish our time together. I also wanted to touch on your sourdough starter study that you recently did as a head nod to so many of our listeners who have started making their own sourdough breads at home. And this falls in line with, the people who also appreciate the yeast that ferment our beers, who help with our wines. But what did you end up looking at with your sourdough starter study?
Yeah, so I will point out that I was just part of a larger team that studied sourdough, so I was helping out with the study. Science is always collaborative, right? And that's what makes it so fun. But yeah, and I will also point out that we started the sourdough study before the pandemic. So we were not just
you were ahead of the curve.
This was not science
You knew
for the
something we
trend.
didn't.
We were ahead of the curve, exactly. And essentially, this was really fun because it was a citizen science, public science, community science, however you want to call it, study where we had people send samples of their sourdough from all over the world, actually. Most from the US, but from all over the world. And we used some of these same techniques that we use in soil, DNA sequencing and other tools to look at the bacteria and fungi, and primarily yeast, of course, in sourdough, to basically see what's out there. And then we did some additional studies, like, does it matter what microbes are in your sourdough? Does it matter for how quickly that sourdough rises and what sort of aromas are produced as your starter is working? So it was a really fun project because we got samples from all over, and they often came with stories, right? This starter was gifted to me by my great-great-grandfather.
It's intergenerational that these cultures have been kept alive to produce the family bread. Yeah, these are heirlooms.
The cultures
I was just thinking about
they outlive
the immortal
people.
jellyfish, and how like jellyfish and sourdough starters, they never die. Self-proliferating. That's Go ahead.
So I'm coming from the West Coast. I'm assuming that there were some stories about the San Francisco starters, because San Francisco yeast and San Francisco is supposed to have special microbes in the air, and it's a very special place for sourdough.
Yeah, and no offense to anyone from San Francisco.
Oh no!
I will say, there was a chat because this was a large team effort, so we were trying to come up with a title for the paper, and I was like, we should call it San Francisco, You're Not That Special. But that got vetoed. But we didn't see if you look in the Bay Area or in San Francisco, all of a sudden you see different sourdough starters. I'm sure there's some magic to the San Francisco sourdough starter, but it's not necessarily in the types of microbes that are in the sourdough starter. But there is a lot of variation. The starters aren't all the same. It just doesn't follow some geography. It's not like, New York sourdough starters are different than California, and then we get this big beef between whose sourdoughs. I mean, there's a lot of variation. And a lot of it's probably determined by what's in your home, where you got your starter from, what you feed your starter, and the list goes on, what temperature that starter is exposed to. And so there's many factors, but there's a lot of variation. And not all sourdough starters are the same. There's a lot of differences in terms of how quickly it rises and the aromas, the compounds that are produced as the sourdough matures.
I think it's wonderful. I love this interaction between our culture quote unquote, and the sourdough cultures, and how they can potentially tell stories about human history and human interaction with these microbes and with our own built environments, and where we've been, and maybe tell a much deeper, richer story about through our food.
And
which I think is
specifically
very
that in our built-in society, because these are people keep these yeast cultures in their refrigerator. How many generations back before you don't have a refrigerator? Like, how are these cultures kept alive? In the cellars, most likely. Someplace, so there was also like a different environment that they were even raised in to be into this intergenerational storytelling. And we've sort of changed that. So that's also got to factor in. Maybe the bread is just not good. Maybe we all have terrible bread today. We just don't know it.
I just think it's great that people have these microbial ecosystems in their homes that are almost like pets, right? Some people
They
even
are
have
like-
names for their starter, right?
Mine has a name.
And
Yeah.
it's beautiful, right? It's like taking a pet rock to the next level. I mean, I think it's just great, and the fact that they're distributed around to family and friends is just great.
These microbes these yeasts have figured out one of the best survival strategies: how to use people to enhance their spread across the globe. I mean, talk
I'm
about a smart
servant to my
species.
starter.
They're piggybacking on human behavior. This is amazing.
Margot would never do that to me.
Margot would never use you in that way. All right, Blair.
Margot the mother doe.
Dr. Fierer, thank you for joining us tonight. I'd love to know, as a last question, if you have anything else about your research or a take-home message that you would like our audience to know about the microbial world around us.
Oh, there's just so many mysteries. So one thing is there's so many mysteries that are waiting to be solved, and also just that not all microbes are bad. I know they've gotten bad reputation recently for good reason, but they're not all bad. Most of them are probably innocuous. Some of them are beneficial, directly or indirectly to us, and we're just at the point where we're starting to explore that in more detail, how we can use microbes to improve our lives, and it's going to be a lot of fun. There's a lot more exciting discoveries to come.
Well, I'm glad that you are a science cheerleader out there and helping the next generation learn about microbes as a career, and giving them a role model of someone who's interested in this area, so they can maybe look ahead and say, I want to study microbes someday. This is what I want to do. The bugs in the soil, but not the one with legs. Thank you so much for joining us tonight. It's been really wonderful getting to hear about your work.
Oh, thank you. It was a pleasure.
And can people find you anywhere online?
Yeah, I'm on Twitter, or fortunately my name is unique enough that nothing else pops up. But yeah, on Twitter is probably the easiest.
Okay, fantastic. Thanks again. Have a wonderful night.
Thank you. Bye.
That might be the first time some of our listeners have heard that there can be bad microbes.
You think?
We talk a lot about
On
the
this
positive.
show?
Yeah, this show covers a lot of the positives of what microbes can do for us. Yeah, there are some outliers. You'll hear about them somewhere else.
I can't wait for, the microbe movie to come out. It's going to be the good, the bad, and the innocuous.
Good.
Anyway, this is This Week in Science. We hope that you enjoyed the first half of This Week in Science and our interview with Dr. Noah Fierer. And if you are appreciating the show, please take a moment to head over to twist.org and click on the Patreon link there. It'll take you to the Patreon community for TWIS, where you will have a chance to choose your level of support to support TWIS on a monthly basis. and help us continue to bring science and interviews like this to you and to other people every week. We really can't do this without you. Thank you for your support. And we're going to come on back right now. This is This Week in Science, but you know what time it is?
What time is it?
Time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have babbling baby bats.
Ba-ba-ba-ba-ba-ba-ba.
Yes, babbling is part of learning speech. It's key to developing careful control over vocal cords and all of the kind of vocal gymnastics that we do to make human language. And so this new study published in Science this week indicates that the same is true for greater sac-winged bats, Saccoteryx bilineata, which are native to Central America. Co-author Miriam Knornschild, behavioral ecologist at the Museum of Natural History in Berlin, says, quote, human infants seem to babble on the one hand to interact with their caregivers, but they also do that when they're completely alone, seemingly happily just exploring their voice. And that's the same that our bats were doing. So in this study, this was in Costa Rica and Panama. In 2015 and 2016, they looked at 20 baby bats, and they were recorded in the wild. These bats in particular are forest bats, which made it easier to study them. They didn't have to stay in a cave full of bat guano and try to hone in on a cacophony of bats. Because it was the forest, they could kind of just identify some babies and mic them up. And these bats, they start babbling at about 3 weeks after birth. They babble for about 7 to 10 weeks until they're weaned. And during this period, they spend around 30% of their days babbling. That is a lot of time, with sessions lasting on average about 7 minutes. But one bat babbled for a full 43 minutes, which is so long, especially considering that normal bat communication, it only lasts a few seconds. So it's very quick. And what's really cool is they took these vocalizations that they recorded, they converted them into images called spectrograms. And the syllables, what they consider syllables in the babbling, had very specific shapes so that they could kind of distinguish them and pair them. They analyzed more than 55,000 syllables, and they found universal characteristics of babbling in human infants and in the bats. So there were things like repetition of the same sound over ba, 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 a lack of meaning, and then also that the sounds followed a certain rhythm. Here's the other thing that's really cool. The researchers also found that some young bats learned fairly early a 6-syllable song that is used by males to mark their territory and attract females. So this is one of the first things they picked up, probably equivalent to, a baby learning how to say da-da or no or something like that. And so the pups listen to the adult males, and then they imitate that song back. So you can see to the naked ear, just sounds like a lot of squeaking. But to adult bats, it's nonsense. And so these pups listen, and then they imitate this short song. And for males, that would make sense because they have to grow up and sing that song. Females also would imitate and learn the male song as babies, but would not then turn around and make that sound as an adult. So the suggestion is that this is part of their cognitive growth where they learn meaning. And so they could be babbling this back to form this song because that's still really important for their sexual selection later in life. So they have to understand the meaning behind this 6-syllable song. And of course, to this point, is always included in these sorts of studies, very few other species babble. Only some birds, some marmosets, perhaps some dolphins or beluga whales. Come on now. Probably most animals babble and we just haven't found out yet.
That's actually sounds like a lot of animals as you start listing it.
Yeah.
Like,
yeah, so
actually, there's a lot.
Yeah. Yeah, and it's all like that list is animals that have social structures and have complicated vocal cues. So, of course, if this is how our brain develops speech, it can't be that different in other species that use vocalizations, complex vocalizations. So I'm sure if we started to look closer, we find more and more species that do this.
I wonder though, so we talk about this in the babbling, it's these repeated syllables, it's repeated phrases, it's the also just kind of playing with different sounds and looping them together and seeing how they fit together. Ooh, oh yeah, yeah, yeah. Oh my god, Gucci, Gucci. Just and babies are going to follow those kinds of sounds and make them themselves. But I wonder if in some animals, say like cats, where cats exist in the wild in prides, where they're small groups, but they do communicate, yet not necessarily in the same phrase and syllable type way that we expect,
right.
that it's a roar or it's a, if there's something even more finely, a finer resolution of detail in the babble, it just that's in the sound itself.
Yeah. Well, and cats are a weird case too, because they meow for the benefit of humans. That's not something, right, that they do in the wild to communicate with one another. So
I don't know. My cats meow at each other. They meow for each other when one is downstairs and one is upstairs.
Well, I'll see if I can find the study, but I think we reported on the show that
did, and I don't
is
believe
for
it.
humans. You don't believe it? Okay. Well, that's a whole other story.
I'm pulling a Justin now.
Oh, okay.
I don't believe
well, that's fair. You can disbelieve stuff. I actually disbelieve the thing that you just said, Kiki, about babies understanding or being attracted. I hear parents doing that all the time. I've never heard a
there are
kid talk
studies
like...
there are studies that suggest that babies like that kind of vocalization
they may
better
enjoy
than
it.
actual words.
There are also studies
like,
though,
I hear that
show
no kid talks like this, but adults seem like compelled to speak in that voice to children, to infants.
Yeah.
There are
Fair
also
enough.
studies that show that the more you speak to a baby in complete sentences like an adult, the quicker they develop speech.
Yeah.
It's complicated. The human brain is very complicated. It's not black and white, but there is a whole movement called talk, read, sing, which is about talking, reading, and singing to your baby from the moment it's born. And it is because it does help with the development of language. So it's anyway, very, very interesting.
And we've seen in birds, this babbling is thought to be practice for the songs as well. Trying out different sounds, seeing how they work, putting them together in different ways. It's interesting to hear that bats do this as well. They don't just squeak at each other; they squeak sentences potentially.
Yeah, and I think the thing
they be babbling.
Yeah, the thing that's really interesting about it too is that I think I kind of glossed over it, but they do this to kind of have a communication moment with adult bats, but they also do it when they're alone. And that's such an important piece because then they are kind of just exploring what their vocal cords can do.
Yeah, I've seen it in infants, being a father many times. There is this sort of right before full speech. There's usually this period of talking in babble sentences where they're going through all of the motions of a full conversation by vocal, and there's not really words, and then suddenly they're talking with the full sentence. It's like a very
You're like,
strange...
wait, did you just ask me how my day was?
Yeah, it's kind of like that. But there is this there's this sort of decision or something that clicks that's trying to put that whole word language thing into a functional as opposed to just a practice. And it comes out as this sort of blither blather for a little while. And then it like it clicks in and they're often talking. And then they just don't stop.
Yeah, they don't. And here we are today, still talking.
Yeah. Yeah.
Speaking of talking, let's move on to talk about some monkeys.
Yeah, you got it. So I have a study about acting under pressure and performing under pressure and how that can be sometimes difficult for monkeys. This is from Carnegie Mellon University, and they found the kind of the liner notes here are that, like humans, rhesus macaques can choke when facing a high stakes situation. That's not choke like you didn't chew your food right. That's choke like you mess up. Like when you parallel park every day and it's easy, you're not even paying attention. But when someone's in the car with you, someone's sitting in the car that you're parallel parking in front of, you have to do it 5 times and then you give up and leave. So it's about choking under pressure, right?
Yeah. Are you talking from personal experience?
Not at all. Prior research, along with anecdotal evidence, suggests that humans are prone at times to choking when faced with a high pressure situation, which in that one case is just social pressure, but in other times have real world stakes. And so this was a series of experiments with a group of rhesus macaques. They trained 3 rhesus monkeys. So these were just 3, and then in the study that the test case was small due to animal welfare rules. So this is a preliminary study. It is something to be considered that it's just 3 monkeys, but they trained these 3 monkeys to engage in difficult virtual reaching tasks on a computer screen. And they knew beforehand the desired treat was available if they got it right. The researchers then set up 4 scenarios for the monkeys: small, medium, large, and a jackpot. And as the tests grew more difficult, the rewards grew larger, and each monkey was tested 10 times. They found that all of them worked harder to get the treats that were higher value as they moved up in difficulty, and the rewards got bigger and they were rewarded as was appropriate. But when it came time for the jackpot, monkeys failed miserably. Both the speed and accuracy declined between 10 and 25% of the time, and that prevented the monkeys from receiving their jackpot. They also noted that the experience of choking did not appear to change strategies over time. They didn't go, okay, next time I'm going to do this differently. They would do it. They would make their mistakes again and again and again. So this is, again, a very preliminary study, but it does appear to show that what we consider choking, failing when the stakes are high, is not simply a human trait.
And it's not just you parallel parking, or it's not just me, or monkey see, monkey do. See no evil, hear no evil, speak no evil.
One of them can't handle the pressure, guaranteed.
One of them, someone. You know, and different people do differently. Different individuals, probably different monkey individuals do differently on different days, given hormone levels or environmental pressures or, just stress levels from prior interactions. There are probably lots of things that go into choking under pressure, even for a monkey. Yeah. Poor monkeys. I'm not going to choke under pressure. I'm going to ask you right now. Do you like This Week in Science? We love bringing it to you. Thank you for being here right now. If you really like This Week in Science, consider heading over to TWIS.org and clicking our Zazzle link and picking up some of our merchandise. We've got some great hats and mugs and t-shirts. I got a beach towel that I'm very happy with just this last weekend. So the Zazzle site for TWIS.org. Highly recommended. Please head over there today and support TWIS and get your merchandise fix. Heading on back right now, Justin. What do you have for us?
Crop farmers, home gardeners, and vegetarians have this in common. They all don't like insects that eat their plants.
Fair enough. Yeah.
So predator insects that feed on the bugs that eat plants emit odors that the bugs that eat plants can smell. And it changes their behavior. It can even change their physiology so that they can attempt to avoid being eaten. Researchers now report they have developed a way to bottle the smell of fear produced by predators to repel and disrupt destructive insects naturally without the need for harsh substances. This is says quotey voice Sarah Herman, PhD, the project's principal investigator. It's not uncommon to use our senses to avoid risky situations. If a building was on fire, we humans could use our senses of sight and smell to detect the threat. There is evidence for such behavioral responses to risk across taxa that suggest prey organisms can detect predation threats. But the mechanisms for detection aren't very well understood, especially with insects. One of the things that's pointed out, aphids, highly destructive, right? Aphids, they show up in mass, they can destroy crops, they're all over the roses, they can attack the tomatoes, they go everywhere. They have the ability to transmit plant pathogens, too, from one plant to the other, so they can even be a vector for plant problems. They happen to be a favorite food of ladybugs, which gardeners will buy frozen ladybugs that they put out into the yard. Most of those ladybugs, by the way, as soon as they come alive, they're pre-programmed to go traveling. They always want to leave wherever they start to go. So as long as every one of your neighbors is doing this, you'll have ladybugs. But the researchers showed that they could use the smell of ladybugs and to expose them to the odor cues given off by ladybugs that would cause aphids to slow their reproduction rates and even increase their ability or their tendency to grow wings, something that they would need to then avoid the threat by leaving the plant that they had found. So the team would set up an experiment also then to try to isolate the specific components, the chemical components within these odors that are actually doing the pest repelling. And they hooked up the antenna of live aphids to an electroantennogram. That's a thing. Now we know. An electroantennogram, an EAG machine, exposed them to individual odors and predators emitted to see which compounds they detected. And once the compounds were identified, the team set out to create a special order blend that could be then used as an essential oil diffuser that will spread the scent over time across, say, a garden, a crop field, something of a much larger context. So that's the next step. But they definitely identified some of the components within the scent of a ladybug that struck fear into the hearts of aphids.
This is how Batman villains happen.
Wait, what?
Scarecrow.
Oh.
This is his whole deal. The smell of fear.
Yeah. Yeah. So what's really amazing about this is high school girlfriend's stepfather, kept winning awards at the Yolo County Fair for his produce that he grew in his backyard. And his secret, which I think it's safe to share now.
No, you can't.
Was that he would collect bugs from the yard, grind them up into one of those little containers, and then spray that all onto the plants.
Fascinating.
Now, to him, it was a warning. This is where bugs come to die, is on my plants, and to create the fear. Yeah. But it could have been the mixed smells of the mixed smell of the predators in there, predator bugs in there as well as the pests, or it could be the scent of dead pests that were. But he was actually intentionally using the olfactory of the insects to scare them off. So that's fascinating. It's
either the scent of fear or the scent of death, right? Yeah.
Two things that are always fun to. Speaking of the scent of death, 1492, Columbus sailed the ocean blue. He sailed from Spain to India, or so he thought. Actually, it was off by about 9,000 miles. It actually landed in the Caribbean, where he almost immediately enslaved the native populations, put them into labor camps that worked people to death, sent them back to Spain as slaves. There was mass murder, rape, torture, and before it was over, it amounted to the largest genocide the world has ever experienced. All this in the name of the Holy Trinity. His words, not mine. Word of what taking
New
place
world.
did reach the old world and got some mixed reviews. They're doing what to the people they found there? All was then seemingly forgiven because the stories emerged about what was happening in the New World before the heavy hand of the holy warriors had arrived and brought civilization was that the natives were engaged in cannibalism. Oh my gosh. Now, of course, whatever needs to be done. Turns out that was not true. However, a paper in Scientific Reports January 2020 made some claims about cannibals migrating into the Caribbean circa 800 AD. And now a group of researchers who more heavily researched these things in this region have come out and rebuked the paper. They're saying that basically there's plenty of evidence that you can see within a record through the archaeological record. Historians, anthropologists, archaeologists can all look through these records and not find a single trace of evidence. Evidence that should exist, that should be findable, is not there. Giovis and 9 colleagues joined forces to challenge the paper's findings, including Florida State University's Tom Leopard, the University of Oregon's Scott Fitzpatrick, a professor and associate director of research at the Museum of Natural and Cultural History. They say they were disconcerted about the revitalization of cannibalistic narrative, which is not only disingenuous and hurtful to indigenous groups, it's absolutely not true.
Wild.
So there is, so they have also published in Scientific Reports their rebuke of what they say was a poorly put together piece of work. And you know, this is a long-standing thing throughout a lot of, not just the Caribbean, but a lot of South America. We've talked about it before. Because the conquistadors, who were murdering, raping, pillaging, enslaving, were reporting that the natives were engaged in barbaric human sacrifice. Archaeologists who would then find what would anywhere else in the world be called a graveyard were identifying it as a site where ritualistic sacrifice took place.
Meal prep.
The evidence, the body arrangements, the all ages, the across different ages, of the family relatedness, all was ignored, which would identify as a cemetery anywhere else in the world. And South and Central America would be identified as a site of ritualistic human sacrifice. So these things still are with us and hard to get rid of. But no, the Caribbeans, in the Caribbean, they weren't cannibals. Although which is too bad, because in the Pirates of the Caribbean, I do like the fact that they were going to eat the chief. But Jack escapes and then make the new chief the dog. And then you realize, oh, wait, now they're going to eat the dog. Okay. New data. This is a new story. Last story of the night.
What? For you.
My last story of the night. Thank you. New data shows political donations from companies like Core Civic and the Geo Group are influencing policymakers to support legislation criminalizing undocumented immigrants. I talked a little bit last week, or mentioned it, I think, in the disclaimer. You got to look for the profit margin behind some policies to understand why they're so entrenched in certain ideologies. These two companies, Core Civic, the Geo Group, operate more than 200 private incarceration facilities nationwide, detaining thousands just in the 5 facilities they have in New Mexico where this research was originating from. Researchers Lauren Collingwood from the University of New Mexico, Jason L. Morin from California State University Northridge, and Rachel Torres from James Madison University looked at the relationship between congressional bill making and the private prison industry lobbying. And they couldn't find any links between policymaking and private prison contributions to campaigns. Just kidding. They found direct links between them.
Of course.
That's what I would think. Yeah.
Their article, Co-Sponsoring and Cashing In, U.S. House Members' Support for Punitive Immigration Policy and Financial Payoffs from the Private Prison Industry. So what we're talking about is a $4 billion a year industry that only increases with the more people that you incarcerate and can put into that prison system. So this is billions of dollars at stake. Private prison industry, of course, has this interest in making sure more people can get pushed into jail for their business to succeed. Interesting, the researchers found private prison companies tend to make large political donations after House members co-sponsor punitive immigration legislation. That is, the private prison industry is sending money to legislators as a reward for supporting immigration policies that put people into their prisons. They found they also increased the financial payoff for members of Congress who faced more severe political risks by supporting the legislation. And according to the data, these politicians are more often turning to co-sponsorship or group sponsorship of these policies so as not to be singled out for the political cost for supporting these policies, making it look like it was just a consensus thing. So anyway, when you look at some of these policies, know that there's real human suffering going on. You know, it's we aren't, and also we just to be clear, a lot of this we're not just talking about people being incarcerated who snuck into a country. We're talking about people who could have been born here, and we're also talking about people who sought asylum, who have found their way into these detention centers, as opposed to being accepted or turned away. So, be wary. There is money behind the policy that is amplifying. I can't even say it's amplifying an ideology. It's just okay. I'm gonna stop.
It supports an ideology, and we need scientific research sometimes, social scientific research, data investigation to be able to put these trails together. This is political science, social science. This tells us the story of who we are right now, why we're here, what we're doing, and maybe what we need to do to change so that we don't do this more in the future. But let's talk about change in terms of brain organoids. Yes. So brain organoids are these wonderful little tiny balls of cells that are composed of neural tissue, nerves, neurons, and not neural tissue, the supportive cells that are astrocytes and not necessarily what is considered the neural tissue in our central nervous system. These balls of neural cells and their supportive cells grow from induced pluripotent stem cells to become a complex neural tissue that can be used by researchers to investigate the development of the human brain. And this is great because you can't just go into a person's head and open it up every 5 weeks to say, what you doing right now, right? We don't do that.
I mean, you could.
It's frowned upon. Indeed. So researchers have been developing these organoids, and the news to date has been very interesting, and some people are scared from time to time of the implications of what this means. Oh, brains in a dish, mini brains, all this. But they're not far enough advanced to have cognitive thought. They're not far enough advanced to be a fully capable brain. And if they were a fully capable brain in a dish, they're not really connected to anything else, usually. So last week, I wasn't here, and there was a cool study that came out that I'm going to get to in just 1 second. But this study out from this week, researchers at the Eli and Edith Broad Center of Regenerative Medicine and Stem Cell Research at UCLA have been able to grow brain organoids from stem cells from people who have a neurological disorder that's called Rett syndrome. And in growing these brain organoids, with normal cells, they're able to see waves of electrical activity connecting the cells, and there are patterns and fluctuations in the electrical activity through the developmental stages of the brain. And so they've come to understand what normal, quote unquote, activation of these little mini brains is. And in this study that they published in Nature Neuroscience, now with these Rett syndrome mini brains produced from these cells that have disorders, everything looks fine on the outside. And this is part of the issue. Rett disorder, everything looks structurally fine, but functionally there is misfiring and misconnection. And so it is disordered electrically. And so the communication in the brain doesn't happen the way that it should. And in this, they were able to actually see the disordered nature of that the Rett syndrome actually causes as the brain cells, as these organoids were developing. And what they're saying is this really implies that what we're going to be able to do is take cells from people with different disorders and really be able to investigate the neurological underpinnings of where disorders go wrong. What happens, even if something looks structurally fine? What's going on electrically? What's going on with respect to ion channels molecularly that is disrupting the signaling within a brain? And potentially, because you've got these brains in a dish, you can try out all sorts of drugs and possible treatments to be able to see if they will work. And in this particular study, they did test a new drug that is in development on these brain cells, and it corrected the electrical activity in these mini brains in the dish. So it gives evidence that this one particular drug that's being developed might be a promising direction to put more resources toward, which is pretty cool. But then last week, like I was going to say, I said, normally those brains in the dish, they're not really connected to anything.
Yeah, I noticed that. It's like she's onto something.
I put little pins in the conversation that I come back to at certain points. Well, the researchers who published in Cell Stem Cell last week, they developed brain organoids, and in their title, they say functional integrated bilateral optic vesicles. So what does that mean? They developed brain organoids with eyes or eye spots.
Mm-hmm.
They developed brain organoids using information from previous studies showing that they could move backwards and through stem cells create retinal cells with retinal pathways, but that weren't connected to any brain. And they realized, okay, well, to understand how the connections happen, the retina usually is an extension of the brain. So what if we take the stem cells and just tell them to produce retinal cells? And so they got it to work, and their brain cells, these little stem cells, grew into little mini brains with eyes that responded to light.
It's so cute.
This is very cute. Yeah, so this allows, again, it's interesting because of the functional aspect. So we've got, once again, one of the strengths in biological research is structure and function. And so here we can see the structure, how the connections occur through development up to 60 days. So this is a very long period of cellular development here, and can get a real picture of how things change structurally and functionally in the brain organoids and the visual system as it develops. But it's not alive, and it doesn't mean that we're going to be creating little people anytime soon. This is just a question of what sight is, and consciousness and awareness. These are all very different questions, but from just the research perspective of what these advancements will allow, the questions that will be allowed to be investigated.
Yeah, and if consciousness is achieved in a dish, then we've really narrowed down what consciousness is, right? We really then we can really point to it and go, aha, now we've figured it out.
Right.
But it's also like to the first story that you were talking about there. You know, the Rett syndrome and everything being sort of structurally intact. If you just Google small cluster of neurons, there's a bazillion stories of things being discovered to be controlled by just a small cluster of neurons, controlling sleep, diet, age, whatever it is. There's just a small cluster of neurons that have the job to run huge body-wide systems, life-wide systems. And that whole those waves, the propagation and everything could just come down to a tiny cluster of neurons that aren't communicating properly or not acting properly. We think of this brain too, and such with like all of these massive connections that are going on in there. But a lot of the actual work is done in a very small business sort of grassroots effort. Little group of neurons.
Little group over here, little group over here.
Yeah.
Everyone's got their own thing that they're doing. Oh, we can maybe we can coordinate. That's cool.
Yeah.
Yeah. Everyone's got their own plans, but if you can get them coordinated, maybe you can make a person walk. There you go. And then my last story for the night has to do with Tourette syndrome, but not really having Tourette syndrome, but having Tourette-like syndrome and having it kind of caused by social media, which is what I found just absolutely fascinating in this study. The researchers publishing in this advanced article in Brain, they are positing that because of the stress in modern society and our connectedness through social media and video and audio platforms, so like YouTube or TikTok, that people are able to connect and there are behavioral memes that begin to spread as a result of this connection. And they have started suggesting a specific term that they are calling mass social media induced illness, in which they and others have reported people coming in with functional Tourette-like symptoms, such the tics, cursing or lashing out or throwing objects, but they don't actually have a Tourette's diagnosis. And in fact, they can track in Germany a spate of cases that all come from one particular YouTuber who does have Tourette's, but has a tendency to potentially, in their words, it seems to overplay the actual symptoms of the Tourette's in favor and sensationalizes them for YouTube ratings and popularity. And what's happened is other youngsters, teenagers, are picking up the tics.
Is it working? What are his likes like? What is the revenue he's got on his stream? Should we be doing this?
Yeah, right. We should. No
Oh
cursing.
my God. This really just sounds like someone rocking back and forth on their porch, yelling about those young kids and their loud music. It just kind of sounds like somebody, like, this is just something that is part of being young now. Is experiencing social media and the things that you see on social media you repeat.
Absolutely. But the repetitions that are happening are leading to individuals going in to get diagnoses but not necessarily getting diagnosed by an expert in Tourette's. And so as a result, say at their high schools or they're not having to do PE anymore, or they're getting out of things that they don't want to do. They're using it as an excuse to get out of activities. It's a fascinating phenomenon, and that they are concerned that it is going to do a disservice to people who actually have Tourette's and actually suffer from these symptoms, in that it's being co-opted by people, and potentially this is a social media induced illness, and we can consider it as a mental illness, not a disorder like Tourette's, but that and what they're hypothesizing is that it is the stress of modern society, the COVID pandemic, all sorts of things, and having to seek attention on social media, get attention, having an identity crisis like we've never had an opportunity to have before because of social media, that it's leading to people seeing these things, and since they are exposed to them, potentially falling into patterns of expressing these behaviors themselves, even though they don't have the illness. But there is an illness that's involved here, but it's not the diagnosed Tourette's.
Interesting.
It's fascinating. I've read this paper a couple of times, skimmed through it a few times, and I'm not completely 100% sold on it, but the psychology that is here is just fascinating.
It's how do we know it's an illness, and it's not just that teens that like need social interaction and socialization spent 18 months at home on TikTok, and so they became less, this is going to sound so gross, but like less socialized. Like basically they didn't have somebody going like, shh, you're being too loud.
Right. Inside voices. Keep some thoughts you're not supposed to express. Keep them in your head.
Yeah.
So
that's an interesting idea too. I don't know.
Not exactly related, but this is the story I didn't bring tonight. Out of the Aston University, says the use of swear words has declined more than 25% in the UK since the 1990s, according to research.
And I was going to curse and I was going to bring up multiple epithets, but this is a family-friendly podcast.
But apparently the British are cursing less.
And that's what one of the things I'm throwing out there is like, was the last generation heavy metal and swearing, and this generation is weird TikTok.
I haven't heard it.
Don't smoke. Don't
I haven't heard it quite as much. I do recall as a youth, and I don't know if it's the same with youth today. I recall quite a few people using what did you call it? An epithet? Epithet? Yeah. Every like third word.
Yeah.
Like some that was like almost like it was like all the words that they had. Like,
yeah.
And you don't really hear, but and then again, I'm also much older. I hang out with a different crowd now. That might be also why I haven't heard it anymore. I don't know. I have to find some young people and see if they know any curse words.
Well, maybe our listeners can bring us the young people.
I know they have words I don't understand. I know
find us some young people. If you're watching or listening right now. I think we made it. Have we done it?
Uh-huh.
Yeah, we have finished this episode of This Week in Science. Another episode down. Another week of the year has passed. Many more science stories have flowed into your brains. I would like to thank people who helped TWIS. Thank you to Fada. Thank you so much for your help with show notes and with social media, helping spread those memes, hopefully not Tourette's-like symptoms to our followers. Thank you to Identity4 for recording the show. Thank you to our moderators, Aaron Laura, Gord, and so many others. Thank you for keeping things safe in our chat rooms. Thank you also to Rachel. Thank you for editing the show. I really appreciate the work that you're doing. And thank you to our Patreon sponsors. Thank you, Pierre Velazarb, Ralph E. Figueroa, John Ratnaswamy, Carl Kornfeld, Melanie Stegman, DeCramsta, Karen Tazi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vagard Chefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Don Mundis, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Hey Arizona, support Aaron Lieberman for governor, Rudy Garcia, Dave Wilkinson, Ronnie Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavis, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, EO, Kevin Perichan, Aaron Luthen, Steve DeBell, Bab Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, you can find the link at TWIS.org. We'll be back, and on next week's show, we will be speaking with Dr. Alok Patel from the Nova PBS Nova Now podcast.
Ooh.
Don't miss it.
Don't miss it. Yeah, that'll be Wednesday, 8 p.m. Pacific time, on YouTube, Facebook channels, as well as the website, twis.org/live.
Yeah, and if you want to listen to us as a podcast, maybe on a long drive somewhere beautiful, just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for our newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in that subject line, or your email will be spam filtered into the deep, cold, dry Antarctic wasteland, and we'll never find it. It won't even be able to survive because nothing can, apparently.
Nothing survives there. Nothing lives there. You can also hit us up on the Twitter where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.